We get NADH right there. So we've made another electron carrier. We broke a bond, liberated an electron, and that was picked up by NAD plus to make NADH. Now this whole process right here is catalyzed by an enzyme whose name I would like you to know. It's catalyzed by the enzyme pyruvate dehydrogenase. And I will write that right here. Pyruvate dehydrogenase. Now the reason that that's an important enzyme is because this is the enzyme that I was referring to when we talked about feedback inhibition. Remember, I told you there was one point in cellular respiration when we've created enough ATP that we don't want cellular respiration to be occurring any longer. And that enzyme that's competitively inhibited by the products of that biochemical pathway is pyruvate dehydrogenase. So what pyruvate dehydrogenase does is it takes pyruvate and it takes coenzyme A, it breaks the carbon off and it sticks those two things together. When it breaks that carbon off, an NAD plus is able to come in and pick up an electron that was liberated to make NADH.
So that whole process is called pyruvate dehydrogenase.
This is the enzyme that catalyzes that reaction. So we've had our pyruvate shuttled in here, and we've made another electron carrier, and that's a cool thing. So this next series of reactions that I'm going to show you, including this one, is called the Krebs cycle. It's also referred to as the citric acid cycle. Those are synonymous, they're synonyms. But we'll refer to it as the Krebs cycle. Yep.
Before we get into that, can you explain where glycolysis happens?
Sure. Glycolysis happens just in the cytosol outside the mitochondrion.
Okay.
Yep. Good question. So let me ask you a question about that. So we know that this reaction occurs in the cytosol, and it's catalyzed by a whole bunch of enzymes that are floating around in the cytosol. Where do the ribosomes live that create the enzymes that are catalyzing these reactions? In the cytosol or in the endoplasmic reticulum? In the cytosol, right? If it's in the endoplasmic reticulum, those enzymes are being pumped generally out of the cell. Okay. So I also told you when we were talking about mitochondria a long time ago, the mitochondria have their own DNA. And you inherit your mitochondria from your mother. Now that DNA is important because all of these enzymes that are catalyzing these reactions are created by mitochondrial DNA ultimately. So what that means is these proteins look identical with our mother's proteins, and they might be quite different than our father's proteins. Okay, so the next thing to happen is coenzyme A, and this actual intermediate is called acetyl-CoA. So this is acetyl-CoA. Acetyl-CoA, acetyl coenzyme A, is shipped to the inner sanctum of our mitochondrion. So it's brought all the way inside the mitochondrion, inside the inner membrane. And this is where the Krebs cycle proper actually occurs. So what happens is there is another intermediate, or a shuttle that's kind of like Rubisco in the dark reactions. There is a 4-carbon molecule, and this 4-carbon molecule is called oxaloacetate. Coenzyme A comes up here, and it drops its 2 carbons that it got from pyruvate off on oxaloacetate.
What was that step again?
I'm sorry?
What was that step again? So they combine or?
So coenzyme A is going to drop these two carbons off on oxaloacetate. And then CoA is going to leave and go back to the intermembrane space. And then what we have is a 6-carbon molecule that looks like this. So 2 of those carbons came from pyruvate ultimately, right? But they were dropped off by coenzyme A. And so if we think about the function of coenzyme A, really all coenzyme A is doing is it's shuttling carbons from sugar inside the mitochondrion. And then once it gets in here, it drops those carbons off on oxaloacetate. Okay? And that's the last intermediate name that I'll give you. But please remember, oxaloacetate is an enzyme, and so we're going to need to remake oxaloacetate eventually. Okay. What we're going to do now is a series of reactions in which we're going to be chopping off these 2 carbons to make some more carbon dioxide and to liberate some more electrons. So in the first step, what happens is we chop off that top carbon, the one that's sticking up, so that we end up with a 5-carbon chain. Now, we've chopped off a carbon, we've broken a bond, so what have we liberated? An electron. So we're going to pick that electron up with NAD+. It does. And then that carbon becomes CO2. You okay with that idea so far? Really, this step looks almost identical to this step, right? I mean, all we've done is broken off one carbon that becomes carbon dioxide and made an NADH. And then we're going to do the same thing down here. We're going to take that 5-carbon chain and break it back into a 4-carbon chain that is not oxaloacetate. But because we broke off another carbon, we know that we're going to make another carbon dioxide. And since we broke another bond, we liberated another electron, so we can make another NAD+.
So this happens twice then?
Mm-hmm. So far, within the inside of the mitochondrion. Okay, so this next step is going to be a little bit confusing, but bear with me. What we're going to get is molecular reshuffling, and I haven't shown you all the side groups here, but there are a bunch of side groups. And we're working back to oxaloacetate. So we start shuffling side groups and carbon compounds around, and in that process, we end up with another intermediate that is also a 4-carbon chain. But because we're shuffling things around, we're able to phosphorylate an intermediate of ATP. What happens is a molecule called GDP which is identical to ATP, but instead of adenosine as the nitrogenous base, it has guanine as the nitrogenous base. Comes along and it becomes GTP. And it does that because there is an inorganic phosphate that is able to be attached at that point. That's PI.
Oh.
Yeah, so it's an inorganic phosphate. Is that oxidative or is that substrate level phosphorylation?
Oxidative.
Oxidative phosphorylation, right? Because GDP has picked up a phosphate from the cytosol. But GDP and GTP are very short-lived. Really, the purpose of GTP is to transfer that phosphate from itself onto ATP. And so GTP loses its phosphate, and it gives it to ADP to make ATP. I know. It's unfortunate, but that's what happens. So let's simplify this as much as we can. All we do is we make an intermediate that can shuttle that inorganic phosphate onto ATP.
ADP cannot do that.
Because ADP does not do that.
ADP.
Yeah. Is this substrate level or oxidative phosphorylation? Substrate level phosphorylation, because we're getting that phosphate from GTP transferred onto ADP. Cool?
Would the GTP and the GDP be oxidative phosphorylation?
Yeah, yep, we definitely make GTP by oxidative phosphorylation, for sure. Okay, then the last step of the Krebs cycle is the last bit of molecular reshuffling. So we're gonna move bonds around and different things around. And we're actually gonna liberate a couple of electrons when we do that. And we're gonna make two different types of electron carriers. So one we're already familiar with. We make an NADH. Then we also make another electron carrier. And that other electron carrier is called FADH2. So we start off with an FAD plus, and we make an FADH2. Functionally, they do exactly the same thing. They just take electrons from one spot to another. Okay? All right. And we've reclaimed our oxaloacetate that can continue spinning the Krebs cycle and making more electron carriers. So let's think about the purpose and the products of the Krebs cycle. So the purpose of the Krebs cycle, I'm going to write it right here, is the complete digestion of sugar to create lots of electron carriers. Okay, so the purpose of the Krebs cycle, again, is the complete digestion of sugar to create a bunch of electron carriers. In the process of doing that, we ended up creating a little bit of ATP as well. But the primary product of the Krebs cycle is electron carriers. That's what's most valuable to us. Okay, so let's think about products now. So the products are what? What did we make? We made a bunch of NADH. And how many NADHs did we make?
3.
We made 3 right here. 1, 2, 3, and then 4 right there, right? So we made 4 NADHs. What else did we make? FADH2? Yep. And how many of those did we make? Just 1, right here. Carbon dioxide, yeah. CO2. How many of those did we make? 3. And that should make some sense, right? Because we started with the 3 carbon molecule, and for completely digesting it, we have to break all of those bonds. So we have 3 carbons that are now represented by 3 carbon dioxide molecules. And then what else did we make? ATP. And how much ATP did we make? Just 1.
So why wouldn't we add the GTP? Is that just because it makes the ATP?
Yeah, it's just an intermediate. We don't actually use it for cellular energy. Good question. What if I told you guys that the actual products were 8, 2, 6, and 2?
Because you have to do it twice.
Yeah, because we have 2 pyruvates, right? And so the Krebs cycle turns twice for every glucose molecule.
Oh, man.
You okay with that idea?
Okay.
So what this means is I can ask questions in a couple of different ways on the exam. I could say, from each turn of the Krebs cycle, how many NADHs are made? And you would say 4, right? And if I said, with the complete digestion of a glucose molecule, how many NADHs are made in the Krebs cycle? And you would say 8. Exactly right.
So it goes through this whole thing twice?
For every glucose molecule. Yep, the whole thing. Because we start with pyruvates, right? So it's going to go through all of that. Are there questions about the Krebs cycle?
And all of that is happening in the mitochondria.
Mm-hmm. Yep. This first step happens in the intramembrane space, and this happens in the intramembrane space. Yep.
Is there any more steps after this?
Yeah, for sure. But there's steps that you've already seen, so they won't be a problem. Did you have a question as well, Sarai? Okay. Linda?
What did you just say about every glucose molecule in there?
So every glucose molecule is going to produce 2 pyruvates,
Okay, that's all.
and so we spin the Krebs cycle twice.
Spin it twice, okay. Thank you.
Yep, you bet. Okay, let's look at our PowerPoint. Okay, so this is the first step in the oxidation of our sugar molecule after glycolysis. And this is where oxygen is actually required. So if we're working in